Major histocompatibility complex. They are a set of genes located on the short arm of chromosome number 6, and they determine the compatibility of tissues between two individuals and are very important when it comes to transplantation. And in humans, MHC is also called as human leukocyte antigen system or HLA system. Coming to immunity, we can have either innate or acquired immunity. As for acquired or adaptive immunity, it is further divided into humoral and cell-mediated immunity. Humoral is mediated by, like when B cells are activated, they get transformed to plasma cells and produce antibodies. That is humoral immunity, but we are concerned only with cell-mediated immunity in this presentation. And cell-mediated immunity is mediated by T cells, and T cells can recognize only those antigens that are presented to them complexed with an MHC molecule, so they are called as MHC restricted. In this figure, it is a simplified figure showing a T cell and another cell with an MHC molecule on its surface. So this T cell can either be a CD4 T cell or a CD8 T cell. A T cell cannot be both, that is, they are mutually exclusive. That is, on the surface we can have either a CD4 molecule or a CD8 protein. So that is, they are mutually exclusive. I have shown it as a CD8 bar CD4 for simplicity's sake. And this is any nucleated cell or an antigen presenting cell, and it is any nucleated cell in case of class 1 and antigen presenting cell in case of class 2. So this is the T cell receptor that is seen on the surface of the T cell here, and this is the MHC molecule seen on the surface of this cell, and the orange is the antigen or the protein antigen that is being presented by this MHC molecule to the T cell receptor. Now what we have to understand is that the CD8 T cells, they will recognize only those protein antigens that are presented to them complexed with MHC class 1. So they are that is CD8 T cell is class 1 restricted, and as for CD4, they can recognize only those that are presented to or complexed with CD or class 2 MHC. So CD4 T cell is MHC class 2 restricted. Okay. We have already told that MHC is a complex of genes on the chromosome, short arm of chromosome 6, and it is also called as the human leukocyte antigen system. And there are three classes of genes: class 1, class 2, and class 3. And class 3 is concerned mainly with the production of complement proteins like C2, C4, factor B of the alternative pathway, etc. And we will not be discussing anything about that in this presentation. We will be covering only about class 1 and class 2. Now MHC molecules, they are polygenic. That means the class 1 itself has many genes like A, B, and C, and class 2 has DR, DP, DQ, and DR, and that is what you call the polygenic. So MHC is polygenic. In fact, it is also polymorphic, and the most polymorphic system in the in case of humans, and we'll be coming to both of that shortly. This is the haplotype, and here we can see the class 1, class 2, and class 3. So class 1 is further divided into 3 genes, A, B, and C. So that is, they are polygenic. Now MHC is also polymorphic. Now what is polymorphism? Polymorphism means there are multiple alleles that can be present for each gene in the population. So if you take the example of B can have multiple alleles in the population. For example, B1, B2, B3, B4 till B200. Similarly for C, C1, C2, C3, etc. Similarly for A, A1, A2, A3, A4, etc. Now to understand that, I'll tell the example of a shoe shop. Suppose we want to buy a shoe, and we are going to a shoe shop, and the shop or the shoe shop can be considered as the population, and that shop will have multiple brands or brands of shoes, and like Nike, Adidas, Puma, etc. So each brand of shoe we can consider as an allele, and as for the position, the locus, we can consider that as the leg or the foot of the customer. So that foot can have only one of those brands, either Adidas or Puma. Once he leaves the shop or once he is going to buy only one of that for his legs. So that means in the shop there are many brands available, or in the population there are many variations of genes available for each, for B1, for B, C and A, but only but at a particular locus for a person only one of that is possible. So this person may have a B1 or a B2 or a B3, but not all of them. So but in the population it is this. So another person can have B5, this person has B1, another person can have B7, but all of them are not going to have the same thing. So that is what you call as the polymorphism. Okay, now we have already told that there are blocks of alleles called haplotypes which are inherited together. MHC genes are highly polymorphic, and it is another property is the codominant expression. What is codominance? We'll come to codominance after a short while, after this is covered. Now this is an example with a father, mother and two children. I have shown only two children for simplicity sake. Now if you look at this, there are two haplotypes here. That is, each person has two haplotypes. As for this father, this haplotype is inherited from his father, and this haplotype is inherited from his mother. As for the mother, this haplotype is inherited from her father, and this haplotype is inherited from her mother. Now they are marrying and they are having children, and we have shown only two children here, and this child 1 has one of the haplotype from his father and the second haplotype from his mother, and the second child has one of the haplotype from the father and the second from the mother. Now you would have understood that there are four possibilities, even though I have shown only two. Another possibility is this haplotype with this, see, like this father's and this mother's, that is 1, 2, and this haplotype with this combination, or this haplotype with this combination. So there are 4 possibilities, and if you look closely, none of the 4 possibilities are same. That means none of the children are really matching with the other, and the funny thing is that none of the children are matching with either of the parents also. So what we have to understand it, so it is to have a compatible genetic loci is a very rare occurrence. That is why it becomes very difficult to find a compatible transplant donors. It is a very difficult thing to obtain exactly similar people, and in fact it may be very almost unbelievably difficult or almost impossible, except of course in case of identical twins. Okay, now this is a codominant expression. Now this I have taken the haplotypes of the father as an example to demonstrate codominance here. Now this A, B and C is the class 1 MHC, and the DR is the class 2. I have skipped DQ and DP for simplicity's sake. And if you look into this case, he has both A2 and A3, one from his father and from his mother, and at the surface of the cell both A2 and A3 are expressed, and none of them, or A2 is not dominant, or A3 is not dominant, or A2 is not recessive, or A3 is not recessive. Both are equally expressed, both have equal importance, both are equally expressing their proteins. The same is with B3, B1, C2, C4, DR4 and DR5. So that means all of them are expressing themselves. So the good thing is that it increases the number of possibilities, or it increases the number of antigens that can be recognized by these proteins. So even though there is a problem of this transplant rejection and all that, the codominant expression, polygenism and polymorphism also helps the MHC molecules or the MHC system to recognize more number of molecules. So that is the good thing about the good thing about these properties. Now coming to MHC class 1. MHC class 1 has HLA A, B and C genes, and they have actually two chains, an alpha chain and a beta chain. The alpha chain is further divided into alpha 1, alpha 2 and alpha 3 subunits, or made of proteins, and another protein called beta 2 microglobulin is also present. And this beta 2 microglobulin is actually not produced in the MHC position. It is produced in the chromosome number. It is produced by a position or a gene that is located in the chromosome number 15, and it is transported to or comes to the means alpha genes and or the MHC genes, and it forms together. Now what is the use of a beta 2 microglobulin is that it will help to transport the MHC class 1 to the surface of the cell. We will be coming to that very shortly. Now MHC class 1 helps to bind to the peptides from the endogenous antigens, and they will present them to CD8 T cells, and it is present on the surface of all the nucleated cells and platelets. This is the image showing the MHC class 1. We can see that alpha 1 and the alpha 2 subunits form the antigen binding variable region, and this is the alpha 3, and the beta 2 microglobulin is this one, and this is a nucleated cell. Now coming to MHC class 2. MHC class 2, it is formed by HLA, DP, DQ and DR, and they recognize the exogenous antigens, and they present these antigens to CD4 T cell. And I have already mentioned the dendritic cell is the best among them, though the macrophages, monocytes and B lymphocytes can also function as antigen presenting cells. And in case of MHC class 2, alpha 1 and beta 1 form the variable region, as seen in this image. The alpha 1 and beta 1 subunits form the antigen binding variable region. Okay. Now coming to antigen processing and presentations. It is already told that the T cells only recognize processed peptides that are presented to them by the MHC molecules. Now coming to the case of endogenous antigens, they are presented by the MHC class 1 to the CD8 T cell. Now if you look here, there is the endoplasmic reticulum, and the MHC class 1 is being prepared in the endoplasmic reticulum. So let that be here, and suppose this is a normal cell, or it can be a normal cell or a viral infected cell or a tumor cell. There are three possibilities, and we'll take the example of a viral infected cell. Okay, so suppose this is a virus infected cell, and this is a viral protein, and the viral protein, this protein will be degraded by the proteasome, and this proteasome, it will be transported to the endoplasmic reticulum by the TAP protein. Now TAP protein is like, it's a transporter, and it's actually called as the transporter associated with antigen presentation. So TAP protein or transporter associated with antigen presentation will help transport this degraded amino acids to the endoplasmic reticulum, and once it reaches there, this protein can bind to the MHC to the variable region of the this is the variable region. Okay, I have shown this as a curve, or to as a means to make it easier, to make that diagram easier, I have just made a curve. This is the antigen presenting site of the MHC class 1. So this protein will go and bind to the antigen presenting variable region, and once that is bound there, the beta 2 microglobulin that we talked about before, it will help to transport this entire system, that is the MHC class 1, along with the bound viral protein to the surface of the cell, and once it reaches the surface of the cell, it will be recognized by a CD8 T cell. The T cell receptor of the CD8 will bind and recognize this. Means once that protein is recognized, the T cell will be activated. And once the T cell is activated, what happens is that if this cell was a virus infected cell, this cell will be killed by this T cell. If this was a tumor cell and this was a tumor antigen and this was recognized by the T cell here, this tumor cell will be killed. But if this was a normal cell, this T cell will not harm the cell. It will be recognized as a self antigen and it will not harm that cell. Now we'll come to exogenous antigens. Okay, now for exogenous antigens, we'll take the example of a bacteria which was phagocytosed into the cell. That is an antigen presenting cell. Okay, and this was phagocytosed here. Let that be here. Okay, now we have again the endoplasmic reticulum where there is MHC class 2, and if you look closely, this is the variable region of the MHC class 2 here, and the variable region is bound by another green substance. That green is the invariant chain. This invariant chain actually will be bound to the variable region, and that protects from binding any self antigens to the MHC class to that variable region. So this invariable chain, this green here, it protects from binding of self antigens. So that is there. So what happens is this phagocytosed bacteria, it will form the phagolysosome, and this phagocytosed bacteria is degraded into amino acids, and this MHC class 2 will be transported to the phagolysosome, and once this reaches the phagolysosome, the acidic pH due to the phagolysosome will cause the invariant chain to dissociate. Like see here, this invariant chain to dissociate from the variable region, and once the invariant chain is dissociated from the variable region, the exogenous protein, in this case the bacterial protein, will be bound to the variable region of the MHC class 2, and after that this will be transported to the surface of the antigen presenting cell, and once it reaches the surface, this MHC class 2 will be recognized by the CD4 T cells, and the T cell receptor will be will recognize that foreign exogenous antigen, and the T cell will be activated. And now what will happen when this T cell is activated? They will begin to produce cytokines, and these cytokines will activate more macrophages and B cells, and the immune responses will be initiated. So that is how the exogenous antigens are dealt with. Now coming to transplant rejection, we have this transplant rejection is due to the differences in the HLA alleles. Now we have to understand there are two pathways by which a graft antigen can be recognized by the T cell, and this whole process of graft rejection takes place. One is the direct pathway and the other is the indirect pathway. In case of a direct pathway, what happens is that the graft MHC bound with the graft proteins will be recognized by the T cells of the recipient, and they'll be destroyed by the recipient's T cells. And in case of indirect pathway, it is just like what we discussed before. That is the graft. Once we have a graft, once a recipient gets a graft, that graft will be treated as a foreign material and or exogenous material, and that will be graft proteins will be taken up by the host or the recipient MHCs, and the graft proteins will be complexed with the MHC molecules of the recipient, and it will be presented to the recipient's T cell, and it will be rejected in that way. So we have direct and the indirect pathway, and I think that is about it for MHCs and transplant rejection. Thank you.